Hello, hello, hello. Is it working? Yeah, cool, wonderful. So we are all here today to see Virj Kan. We are very happy to have Virj with us today. Virj, you are MIT alumni, Media Lab. You were at Tangible Interfaces Group, no? And also Media Didn't Matter, okay? And you did work a lot on the biomaterial and the biodesign in general. So thank you for being here with us, and the floor is yours.
Hi, thanks for having me and thanks for coming. So as previously mentioned, I am a graduate of the MIT Media Lab, where I worked on a lot of research in the field of human-computer interaction and also biodesign. So today's body of work, I'm going to share a little bit with you some of the stuff that I did in grad school, but then also what I'm doing now today. Today I'm more transitioned my career into the realm of startups and the realm of building a company. So I have a materials company that's based in California in the Bay Area, where we work on different environmental problems such as kelp conservation and also waste reduction. So I'll share a little bit of that. It's all in the realm of materials. But in the very beginning, I will start with a few projects that are research oriented, and then I'll move into more commercially driven projects, and then I'll finish with some art projects that I've been working on lately. In general, I'm interested in just largely how humans interact with the world of information, especially information that is too abstract or too small for us to fully comprehend and understand. I'm interested in building systems that allow us to interface with those environmental information, essentially. And because my goal is to reshape our relationship with that information and ultimately with the environment and our relationship with each other. And so you'll see in this series of projects how I do that through a series of installations, through research papers, through developing technology, and also through making interactive artwork. Just a little bit of kind of starting point. I don't know if anybody knows what this is, but this is a proxemics map. So it's describing the perceptual range that, depending on spatial space or spatial distance, we have a slightly different relationship with the people that are incorporating in that space, or the environment, or the objects. And for me, I'm interested in the sensory system, the human sensory system, and how the field of proxemics can describe our ability for our sensory systems to extend within those different spatial realms. So, for example, within 1.5 feet, it's an intimate space realm. Within 4 feet, it's personal space. And this also describes how you might engage with other people. This might also involve, for example, like a toothbrush that is often in your intimate space. You may not engage with a toothbrush necessarily in a social space. I mean, unless you're going to a hostel and someone accidentally borrows your toothbrush. But and so I use this framework to describe a lot of my projects and the way that I think about my body of work. And as mentioned, I'm only going to talk about a few projects throughout this proxemics. It's a framework described by Edward Hall. And I'm going to talk a little bit about our relationship with our body, some projects that relate to our relationship with objects around us in the intimate and social space, and then as it relates to community and also the environment. And specifically, I'm interested in how we interact with our identity through a social environment, how we interact with climate change or larger environmental systems in the broader space. So first, this is a research project called Dermal Abyss. We published this in the Symposium for Wearable Computers, and then we also got an award at South by Southwest with this project, with collaborators Katya, Nan, Ali, Nick, and Shin. And essentially, in this project, we're transforming the body as an interface for health information by incorporating sensors into a tattoo. So the tattoo becomes sort of a display mechanism, if you will. And the idea is that traditional tattoo inks are now replaced by biosensors. And when you incorporate it into the dermal skin layer, you can then sense different aspects, such as sodium or glucose or different information within that interstitial space to communicate information to your body. So this is a proof of concept tattoo project. There were some challenges in kind of executing this project. So we did preliminary evaluation on like an ex vivo pig skin model. But if we were to extend it further into human skin, some of the unique challenges might be like, for example, how do you know if you're sensing the external environment or the internal? How do you know if it's not something you're sensing in a localized region of the body versus in the global region of the body? So these are some open research questions that we discovered when we worked on this project. If you're interested, you can also find our paper. If you just Google dermal abyss, you'll find it. This next project is called Biodesign. So this is in collaboration with Virj Kan. We published this and showcased this at IGEM, which is the International Genetic Engineered Machines Competition. And they're called biodesign, where we basically made a custom microbial record where you can basically inoculate different parts of your body onto a EP style kind of record that you can incorporate it into a turntable, and you can play the sound of your body. And the idea is that historically music is recorded and stored in a variety of forms. If you look at the vinyl record, it's actually stored in these small engravings on a piece of vinyl material. And the idea is if we utilize biology, we can incorporate music or make sounds that can grow over time, because microbes can grow, right? So you can have dynamic music that changes. And additionally, this can give you kind of a sense of be able to have a different sensory experience with information that you're inoculating. In this case, we're inoculating different parts of our body. So the system is composed of three main elements. So there's an incubator that is basically a turntable that is retrofitted into an incubator, a custom biota record that we made out of acrylic, essentially, and a camera and microcontroller that is pointing right at the record. That sonifies the growth. And as I mentioned, we're specifically focusing on the human body. So what you might expect is a variety of different microflora. Depending on each part of your body, there's a different community of microorganisms. And the idea is that you first start with the user experience, similar to how a traditional turntable DJ interaction is. You basically plate it and incorporate different microorganisms onto the plate. Then you incorporate that plate onto an incubator. It grows over time. And over several hours to days, you have the resulting record that you can then listen to. I mean, let's see if this will work. If I can quickly play this clip. If not, you can also play it on your computer. Anyway, oh, I don't have internet. It's okay. We'll keep going. You can listen to the symphony. But essentially what this website will take you to is allow you to listen to individual parts of what feet microbial colonies sound like, genitalia, belly button, armpit, mouth, and then you can also listen to all together in a symphony. For the way that we're sonifying this is we're basically breaking up each area of the record by different parts of your body. So you inoculate, you apply to different parts of the record, and then the camera just captures an image. The image just has an image processing algorithm, translates that into sound, vectorizes it, clusters the colonies, and then these Cartesian coordinates assigns each colony to specific musical notes that then plays the music from the center. Also, feel free to ask questions if anybody has throughout. So this next project, I'm just going to go through lots of projects really quickly to just tease your imagination, and then we can also have Q&A. You can ask me them. But this next project is my thesis work from when I was in graduate school. It the idea is to, they're called organic primitives, and the idea is to translate basically chemical and biological information into things that are human readable. So what is human readable? Usually we read the environment through our sensory system. So things like smell, our visual system, or texture. So the idea is to create a material interface that allows us to understand what the chemical and biological system is telling us. And typically, the chemical and biological world is mediated through information flows of fluids, like liquids, right? And containing within liquid is like information around pH, information around temperature, information about dioxin content, which gives us this rich body of information about that environmental system that might be inside your body, or it might be in the rain, in your tears, in your sweat, in these different fluid-based systems. Problem with current like human-computer interaction systems in HCI is that you often cannot really easily attain this information or use it in your projects. You can maybe incorporate a microcontroller, but actually the richness of chemistry and biology, you have sensors that are much smaller that allows us to incorporate into materials. So this is what we did, is to basically make a series of these materials that can change color, release odor, and change shape based on chemical information. In this case, it's pH. And we've kind of shown a variety of different form factors like fibers or films, and even a single film that can change both color and shape at the same time. And this allows us to have a tool set of stimuli-responsive biomaterials that you can use to now interact with the natural system with. And the way that the idea is like that it was made is we incorporate dopants into a substrate. In this case, it's a bioplastic, algae-based bioplastic. Some of you have already used with a crosslinker. And then the input is the fluid. And then when you incorporate that material into an object, and when you look around us, there's actually everything is made out of some material. So the premise is that if you replace some of those materials with something that is interactive or stimuli responsive, you can have an object that can sense and display information. So that's sort of what we did. Oh, actually, let me see if I think I skipped a video here. It's okay. But yeah, we made 3 different demonstrations of projects. The first one is this idea of interacting with humans and food. So if you incorporate it onto an apple, the apple can tell you when it's been contaminated during transport. So it displays into like these red spots. Or an umbrella that bleeds in the presence of acid rain. And then the last one is you can interact with plants by incorporating a coating onto the plant, and the plant will then change color and display some information. Let me scroll faster. So this is the library of different materials that we made. We call this the material primitives or organic primitives. In the first kind of column, you get base kind of material primitives that change color. The middle one releases odor. You can turn the odor on and off at demand. The last one changes shape, slightly different angles. And then when you combine multiple primitives together, you can have a single primitive that changes into multiple colors and also release odor, or changes color and shape, or odor and shape together. And then also, while incorporating to the different form factors, you have capabilities to fabricate them into different types of products. And then lastly, you can incorporate an additional level of complexity with computation by incorporating logic, right, into it. So in this second case, by understanding the diffusion rate of the material through with a pH reactive input, you can have a time-telling device, or a device that can titrate across multiple flavor profiles at the very last one. So this is just showing different ways in which you can combine these material primitives together. As I mentioned, sort of how this is done, this is an example of the apple that changes color to tell you that it's been contaminated during transport. And the idea is that a crucial aspect beyond just the sensor development is also the idea of semiotics, creating a biosemiotics that can, why this apple actually can communicate that it's, contaminated during transport is because we understand this Amida muscaria symbol of the poison mushroom, right? And we kind of innately know this to some extent. We know that this symbol means poison. And so by abstracting and extrapolating this semiotic into, say, an apple, you can have a display that you can understand. I'm not going to go into the semiotic theory side of it too much. I'm going to scroll through because I kind of mentioned this series of materials already. So here's some examples of different types of interactions that you can have with, for example, a plant. And because a lot of these, you can imagine it's interacting with fluid-based system, you can transform a plant into a display or incorporating tattoos that tell you information about the external environment by changing color, or shape-changing pasta that tells you when it's ready and done, and an umbrella that bleeds in the presence of acid rain, tell you, ouch, the sky's hurting, or a perfume that by applying the perfume, it changes colors, makes new designs in your clothing, or an apple that tells you it's been contaminated. And finally, a fork that just wilts because you've eaten too much. It's done. So with the example of the mushroom, it's communicating toxicity, but in the case of the umbrella, you're seeing blood, and blood is actually a very innate sight. When you see, an animal also sees blood, and they can understand. It's a very innate symbol. And the further one is this idea of the gesture of refreshing something can also translate to the action of refreshing the new design in a t-shirt. And then a wilting fork to communicate that. And you know the wilting fork from the wilting flower when it's dying and complete. So this body of material, or these organic primitives allows this kind of new open space for design, which I call molecular design interaction. And it allows us to interact with humans and the food system, humans and the environmental system, as well as humans and health information. But I mentioned a lot of this, it doesn't really need to require electronics, but if you want to incorporate electronics to make the interaction go faster or to have more precision, you can incorporate other electronics into the mix. So this is an example of a system that we made that deposited different pH fluids to activate a material. As you can see, within the material system, you don't really need all this bulky electronics. You can just use the material. But the downside of that is just it's 1 or 2 times use, right? It's more catered towards biodegradable materials. But in this case, you can use it for fabrication. You can use it to activate. So this is an example of a project that is exploring kind of using these electronic systems to produce the film, but also activate the film. So this is an example of it producing an edible film that you can then incorporate into a drink. And then there's flavor compounds in the edible film that when you place it into the drink, it changes color to tell you it's all diffused inside of the drink and it's ready to drink. So ways to interact, there's global, local, discrete ways to kind of deposit the fluid and interact with the system. And you know what, I'm gonna skip all this because it's a lot in this one project. But yeah, and essentially that was my graduate research project. Now I have a company. The company started out where we wanted to incorporate some of these materials into solving really big environmental problems like climate change. And one of the big issues with climate change is waste, food waste. And actually, food waste and plastics pollution are interlinked issues because spoiled food is often encased in plastic in the case of store-bought meat, which ends up in landfills and produces lots of methane. And so and also that methane is so much that it's 1/3 of global food produced is lost, which amounts to 4.4 billion metric tons of CO2 a year. So we sought out to kind of address two issues initially by creating a multi-layered material system and a packaging system that can let you know when food is still good to eat, so that you don't produce the waste, and if it goes wasted, you can divert that waste from landfill to compost. And the film is made from multiple layers of different biomaterials, including algae and cellulose materials. This is an example of leftovers that kind of tell you how old it is when it's been left in the fridge, how many days has elapsed, or fish that communicates how long it's been defrosting for by changing color, because over a certain threshold of time temperature, it's unsafe. Or packaging that's sent to spoil to let you know, okay, you can eat this. Actually, no, this is rotten. You cannot eat anymore. And as I mentioned, this idea is you can, if it actually finally goes to waste, because the goal is to actually divert it from going to waste, right? So that you can know, you don't have to guess that it's no longer, whether it's good to eat or not, you can know that it's good to eat, so you will then transform it into energy through your own body by eating it. But if it doesn't, if it's no longer good, then you can compost it without needing to depackage. The whole system can be put, and then we can produce more food and more soil from it. And the technology was, inspired by the early research that I did at the Media Lab at MIT. And the feedstock initially was using seaweed, because seaweed contributes a lot to carbon sequestration. So, and it's also essentially like a carbon neutral feedstock. And we were producing two different products, like a film and also a sensor. I'm going to scroll through really quickly to kind of these different types of technologies, but we tested the material to assess the performance of the film. So in this case, it has UV protective properties that are comparable to SVP 30, and also has a heat sealability and et cetera. But part of the material science and materials engineering is you kind of have to produce tons of these plots for people to really believe you. So hence the plots. Right now we're no longer working on this technology because we discovered actually in California, kelp and seaweed is not actually as abundant of a feedstock. And I'll share a little bit in the next project what we're actively doing now. But a lot of the central to our research development process and addressing environmental issues in particular are trying to address issues at every single point of this life cycle of the product. So looking at the energy costs, the material costs, all of the different attributes of starting from the raw material to the production to when you're at the retail, when you're using it, and also end of life, and thinking about where it goes. And in addressing every single stage, we can maximize the greenhouse gas reductions for the material and the product. So this is getting into closer what we're actively doing now in our company at Primitives. We're working on making a variety of different materials in order to address a key environmental problem in California, but also all around the European coast, including France, Spain, and Norway, which is these sea urchins. And you're probably wondering, like, really? I've never heard of sea urchins being a problem. But interestingly, sea urchins are a major force, a major source of issue for kelp forests all over the world. And kelp, in other words, seaweed, is something that we're exploring in the past, but now we've shifted into using sea urchins because these are destroying kelp forests, and kelp forests are important because they, as I mentioned before, they play a key role in sequestering carbon from the atmosphere, converting CO2 to biomass, but also kelp are the forests of the sea. So with no forest, you don't have fish, you don't have invertebrates, you don't have macroalgae, you don't have wildlife at all when you cut down the forests of the sea. So the idea is that we've been working with the Nature Conservancy, which is a big environmental nonprofit, to remove these sea urchins so that we can reforest the kelp. And there's research papers from the field of conservation that's been studying how to reduce sea urchin, they call them sea urchin barrens, how to reduce them. And one of the really viable ways is actually just to remove them, which is simple enough. And that's because kelp is one of the fastest growing crops in the world, which make them really great for something like bioplastics, but also for conservation purposes, it's easy to restore and recover. So we've been working with sea urchin scuba divers. I'm also a scuba diver myself. And we've been removing the urchins and trying to create a market-based solution to remove them. So we've been making these additives and materials from sea urchins. And the idea is we're kind of using these sea urchins as a biofactory to grow the pigments and the biominerals that can be used for plastics coatings in the building material industry. Because capitalism is a very efficient tool at extracting and removing things from the environment, we're using it to temporarily remove sea urchins to restore kelp forests. And we're doing that by producing also 4 different materials. One is we've extracted a unique class of pigments, pigments and also these composite materials that we made from sea urchins, as well as flexible films and 3D printing filament. The composite materials have comparable tensile properties as natural marble, so there isn't a huge decrease in performance. For example, when we are making sea urchin-based engineered stone. And then also they're fluorescent under UV light, which gives you additional abilities for interaction. And there's a unique class of pigments that are extracted from these sea urchins that give us 3 different colors. And then we also made 3D printing filament that's printable with standard 1.75 millimeter nozzle. So you can print them in basically any of the machines you have here. And our flexible films are also the additive can enhance the processability, mechanical, and moisture barrier properties of flexible films. So essentially that's this project that we're now working on. That's a company. And I mean, I'm actually in France because we're working on potential corporate partnerships to incorporate some of those materials. I'm going to take a moment to just take a pause and ask some questions, because we're going to switch gears into a very, very, very different headspace of projects. And as you can see, there's a knife mounted in the bathroom. You're probably wondering a lot what's happening. But before I move there, any questions? Yes. Sorry, for which project? So there's no mushrooms. It's using macroalgae, so different types of seaweeds, essentially. Like red algae, brown algae, yeah. So we're not using mushrooms at all. Sorry, can you speak up a little bit? I can't hear you. Oh, okay. Yeah, the Amanita muscaria is, so I'm drawing from the symbolic aspect of the poison mushroom that we know from Mario. From Mario and also from popular culture. Because also those animals have evolved this, it's called aposematic signaling, the signaling, this pattern effect to tell us that it's poisonous, don't eat me. So I'm drawing from that into these projects to allow people to communicate when an apple is contaminated as well. Yeah.
But in Mario, it's good to take the point.
Well, yeah, right. Yes.
For the searching stuff. For the searching, I have a question. Do you mix the already existing composites with the searching extract, or is it fully made of searching waste?
It's a mix. It's a mix. Depending on the material pathway, there's anywhere between very little, 1%, up to 90%. and it's highly varied. And part of the idea is you got to balance the mechanical properties, moisture properties, or whatever other properties you want for the use case, the application, but also from like a sustainability and environmental perspective, we are still in the active research as to how much sea urchin we should remove from the environment to restore the kelp forest. So part of it is to enable that flexibility so that we can remove more versus less, depending on the product.
And so the plastic film is made out of plastic and sea urchin, or is it not plastic and another?
Yeah, so there's a variety. So one example is completely biodegradable, bioplastic, but there's another example which is like a composite with a non-biodegradable. And like the idea is depending on what industry you want to go into, you don't actually want everything to be biodegradable. And then also some companies are already using, for example, not biological calcium carbonate, but mined calcium carbonate. So that will allow us to quickly make a shift and also restore the kelp forest and have a carbon sequestration benefit. And it's good for a short-term solution, whereas the biodegradable plastic and bioplastic, to scale that, is more long-term. And I think we need all of those things in short-term and long-term. Yeah.
And how hard is it to melt 90% urchin-made plastic in a 3D printer?
Oh, that's very hard.
Yeah, is it possible?
We could talk about it after this.
Okay.
We can think about it. We haven't tried for 3D printing. 3D printing, most of the 3D printing filaments, they don't have that much bio content in them in general. It's because, you can't, otherwise you can't print from it. Okay. And you really have to tune the particle size distribution to make it like perform in the way that you want. So there's a lot of like tuning that is needed depending on the product.
Thank you very much.
Yeah. Yeah.
So apart from its coloring feature of the sea urchin, like its unique color, what are the other features that you found from your company? What can it be used for as well?
Yeah, so actually one of the interesting things it's currently being used for is it's used as a drug for like eye diseases. So there's interesting like pharmaceutical applications for this that people have been researching. That's one of the aspects. For us, we're primarily interested in replacing the minerals. We're interested in the different minerals. And there's like magnesium, there's calcium carbonates, there's different types of minerals that we're still in the process of understanding more. Yeah. Okay, so I'm gonna move into art projects headspace now. So this is a project called Sushi Factory. So this is done, I have a media art practice. This is done in like we're an art collective called like a defunct collective. And essentially our collective is exploring like how culture, science, and industrial processes interact with the environmental and food system. This is an example of a site-specific installation where we transformed the bathroom into a site for food cultivation. So you have like a tank in the tub, and it's a bit conceptual, but the idea is like to elicit an emotional experience when someone is walking into the space, and how it relates to agriculture and how like the fishing industry is affecting, and because usually the bathroom, you go in there to expel waste. You don't think about food, even though that is literally the way that you transform food, the human body. And also living in a modern society, you don't often have like the opportunity to interact with the ocean or the environmental system. Largely, you interact with the environmental system by going into the grocery store and buying something that is coming from the ocean, or in this case, you're expelling waste that and eventually ends up into bodies of water. And so this is sort of opening this question and this dialogue and this inquiry of thinking about the bathroom as beyond a site of waste, but also a site of food. And so we have basically a robotic knife that is mounted or placed on top of a bunch of dead tilapia, so in a bathroom setting. And I mean, if you look into tilapia a little bit, tilapia is one of the like most widely farmed fish. I think it's the first to second or third large, like most widely consumed fish. And the reason why it is because it's able to be hyper packed in a very small space with lots of disease and polluted waters and still be able to survive. And so if you imagine that this is why our food system is what it's optimized for, it's optimized for organisms like this. And this is sort of, if you go into the exhibit, this is on the left is a bathroom. If you sit on that toilet seat, you'll see a sushi menu for you to look at. Anyway, that's a little bit of like art project for you. This next project is called Life Support. So it's an interactive installation that is, I'm kind of just gonna play it for a few minutes. I noticed is that it's really loud and annoying. But the piece is called Life Support, and it's kind of contemplating and thinking about what the state of biology and the state of science is currently in the way that we think about what is considered alive and what is considered not alive. And I was thinking about this during COVID in particular because many people were inside of these ICU facilities, and they're primarily supported by a machine. And in a way, like our medical system and the current state of science thinks about the state of life and living as humans as being a machine. And if you can connect enough of these systems that will keep your like a living thing breathing, then you're keeping them alive. But it's a little bit of a critique. This is an installation that basically explores artificial life form. It's an artificial life form that breathes through an electronic system that forces it to breathe. So then can you consider this like a life form, right? If this is the state of how our society and culture is thinking about things. Okay. Another art project, this is more a performance piece that is activated by participants. So if you imagine going to an exhibit, you see this little children's table. And again, this is somewhat of an exploration of like a time of COVID and the times that we're living in right now, or post-COVID. It's hard to know, pre, post, are we still in it? But the idea is to explore like the state of being, like our sense of control is lost, and we're placed into this physical state of infancy again, where we're not moving very much and we're just staying put in one spot, and our biology is scrutinized and assessed. And so the piece it basically invites participants to donate and contribute a piece of themselves to the piece, specifically their saliva, containing their microbiome and many different microorganisms. And 32 strangers will basically contribute their saliva to the piece, and they're asked to first, like, spit into like a container, and then provide a signature onto the container to the right, and then they're asked to select a isolated location in which their saliva and their microbes are going to grow, and then they're to inoculate that particular region of the checkerboard. Once 32 strangers are collected, a game of checkers commences between me and my father. And actually, me and my father are a bit estranged. A little bit of personal information. I haven't really talked to him for like 4 years, and this is the first time we play together a game of chess, or checkers rather. And a lot of this performance is thinking about this relationship between the stranger and somebody that's a friend, somebody that's in the community, somebody that is part of the family, and someone that is not. And in this case, you know, my father, there's a cultural barrier, language barrier, because I grew up in the US, he grew up in China. We cannot speak very much to each other, so we don't. And this is a little bit of using this game to bridge that gap, but at the same time, playing this game on top of a sea of checker saliva of strangers, because in a way I have more relationship and connection to these strangers than to my own father. But yeah, the microbes are collected and they're still growing right now, but I'm growing the microorganisms of these 32 strangers, and I have a piece of them in my art piece. This is the last piece that I'll show in the talk. It's called Microbial Portraits, and it's thinking about examining identity a little bit and how identity, like in a way, there's parts of us we can never fully know. This is an example where this is a time lapse where I grew the face microbiome of 2 people, and over time it grows, and you'll start to see, like, depending on the region of the face, you have a slightly different culture, different microbial colony. And this is sort of reflecting on this idea. You can know yourself, you can know other people, but there's parts of you you can never fully know, such as these individual communities that are living on your body. Anyways, thank you. Thanks for your time. Thanks for sharing space. Yeah, questions, comments, inquiry, thoughts?
No question? You gave everything before? You had a question before, no? Not anymore? Yeah, Nicolas.
Aren't there a bunch of laws regulating the culture of human cells?
Sorry, what?
Aren't there a bunch of laws regulating the culture of human cells?
Are there regulations? No. No, no.
For the bacteria warfare project, did you think about like making a literal like warfare between the bacteria, strangers, like making them like trying to colonize, and like a go game, but like with bacteria trying to take like the other space of other bacteria,
Yes.
and having like a
That was one of the ideas, but then from like a feasibility standpoint of making the show, I couldn't have enough time to do something like this. But you should make a new iteration. You should do it. Yeah.
Hi. I had a question about the tattoo project, Abyss. Did you have the chance to test it on anyone on the real skin?
No, no. But there's a lot of people that email us every day asking, like, when are you going to go to clinical trials? I'd like to be a subject.
Yeah.
Yeah, no, not yet.
Okay.
Yeah.
So I was wondering how maybe the body rejects.
Yeah, we don't know.
Okay.
Yeah.
Hello. Did you have an example? Do you have an example of the music played by the cellars with the DJ?
Yes, we do. If you go on the website, you'll see, because I don't have internet here yet. Yeah, I could go to that. Let me see if Biota Beats.
Other questions?
Oh, shoot. right here, if you want to go. You can listen to it.
Are there other questions or no? Okay, well, thanks, Virj, for your talk, your presentation. Virj is going to stick around with us for a couple of minutes again, so if you have questions that you want to ask her directly, please feel free to do it. And that's it. See you next time for the next conference here. Thank you.
Thank you.